First one on the list is the alkane, the baseline structure.
The alkane, yes. It represents a carbon chain that is completely saturated.
Saturated. Let's define that. What does that mean in practical terms?
When we say saturated, we mean every single carbon atom is bonded to the maximum possible number of other atoms, hydrogens, or other carbons.
So all the seats are taken, so to speak.
All the seats are taken, exactly. And critically, all the bonds connecting them are single bonds.
And the naming is the easiest entry point into nomenclature, right? They end in selene, like ethane, propane.
Methane, exactly.
Now, in terms of molecular personality, what does this saturation mean? They seem, I don't know, a bit chemically dull.
They are, in a way. They're like the molecular equivalent of plain steel, scaffolding strong, rigid, but highly unreactive. They're nonpolar, which means they're hydrophobic. They hate water. And that makes them critical for providing what we call lipophilicity to drugs.
Lipophilicity, that's fat solubility.
Right. And that's vital because it allows a drug to pass through the fatty lipid bilayers of our cell membranes to actually get to its target inside the cell.
So even though they might be chemically inert, they are absolutely essential for the physical journey of the drug through the body.
Precisely. They are stable, inert structures. They don't typically participate in binding except through these very weak, very transient interactions.
You're talking about van der Waals forces.
Exactly, van der Waals forces. These short-range forces that attract molecules to each other. But even these weak forces, when you sum them up across a long alkane chain, they can contribute significantly to binding, especially in the hydrophobic pockets of receptor proteins.
And crucially, I imagine they're not easily broken down.
They're rarely targeted by metabolic enzymes. That makes them the most stable component of a molecule, usually.
But the moment you introduce a double bond, that stability is challenged and everything changes. That brings us right to the alkene.
Right. The introduction of a double bond causes what we call unsaturation. The formula is R1, C, double bond, C, R2.
And now the geometry is restricted, isn't it?
Severely restricted. Instead of that flexible tetrahedral geometry of the alkane, the carbons in the double bond force a planar, flatter geometry on that part of the molecule.
And the naming switches to ethene, like ethene or hexene. Now, I know that double bonds introduce a major constraint that really matters in biology.
Yes, and this restriction in rotation is a massive issue. It leads to potential geometric isomerism, what we often call cis-trans configurations.
So the molecule can exist in two different shapes that can't easily switch between each other?
Exactly. Imagine a molecule has two possible non-interconvertible shapes just because of where the R groups are sitting relative to that double bond.
And the body's receptors, the biological locks, are incredibly specific.
Incredibly. They might only accept the cis key and completely reject the trans key, or vice versa. So designing drugs where that double bond holds the perfect shape is a critical step.
And chemically, what does that double bond do to the molecule's stability compared to that inert alkane we just talked about?
It creates a site of increased electron density. It's a reactive center.
Okay.
Those pi electrons in the double bond are much easier to attack by electron-deficient species. So in terms of drug stability, alkenes are inherently more reactive than alkanes.
Are they targets for metabolism?
Oh, yes. They can be targets for oxidation in the liver, and that often leads to the formation of epoxides, which, as we'll see in a bit, are far more reactive and potentially toxic. So alkenes are less stable in a biological system.
And if we take that unsaturation to its theoretical maximum in a simple chain, we have the alkyne.
The alkane contains a triple bond, R1, C, triple bond C, R2. They're named with the suffix -line, like ethine.
And a triple bond forces that entire section of the molecule to be what? Perfectly straight?
Perfectly linear, a 180-degree geometry.
Right.
If an alkane is a flattened structure, an alkene is like a stiff molecular rod.
And I assume it's also very reactive?
Exactly. Because it contains 3 bonds between the carbons, that region is highly electron-rich, just like the alkane. It's a potential reactive site.
Are they common in drugs?
They're less common than alkenes, but when they are present, their linearity is often used to lock a certain shape into the molecule or to provide a very specific, highly localized reaction site.
Making them significant targets for enzymes.
Exactly, for cleavage or reduction.
Okay, that covers the pure carbon-hydrogen backbone. Now, let's introduce the first type of heteroatom. So an atom that isn't carbon or hydrogen, that truly alters the electrical landscape.
We're moving into the halogen heteroatomics.
Specifically, the halowolcane.
Which is a simple substitution of a hydrogen atom for a halogen. Rx, where X is fluorine, chlorine, bromine, or iodine.
The name is straightforward, chloroethane, for example. But the structural implication, you said, is massive. How does swapping a simple hydrogen for a halogen change the whole molecule's personality?
It all comes down to electronegativity.
Right.
Halogens, especially fluorine and chlorine, are highly electronegative. They're electron greedy. They pull electron density away from the carbon atom they're attached to.
And that creates a dipole.
A significant dipole moment, yes. The carbon becomes slightly positive and the halogen becomes slightly negative. The molecule is now polarized.
Which makes that carbon atom susceptible to attack, turning a totally inert alkane into something potentially reactive.
It does. But the nature of that reactivity depends entirely on which halogen you're using. And in drug design, this is a critical distinction.
Fluorine substitution is incredibly common in pharmaceuticals. So why is the CF bond so popular? That's the core question, right?
It really is. I mean, if chlorine or bromine create this big dipole and potential reactivity, why does fluorine seem to be the preferred choice in, up to a third of modern drugs?
Yeah.
Well, fluorine is the smallest halogen. It's almost exactly the same size as a hydrogen atom.
So you can swap it in without changing the shape.
Right. It's a stealth modification.
Yeah.
It allows the drug to still fit into its receptor.
Okay, that's one reason.
And the second reason, which is most important for stability, is that the CF bond is exceptionally strong. Stronger, in fact, than a typical CH bond.
And that extreme strength prevents the drug from being rapidly metabolized.
Exactly. By oxidative enzymes, particularly the cytochrome P450 system in the liver, fluorine substitution can extend a drug's half-life and efficacy significantly.
But wait, if the CF bond is so stable, doesn't that make it harder for the body to ever get rid of the drug? Could that lead to buildup or toxicity? What's the trade-off?
That is precisely the tension in fluorine chemistry. It's a classic trade-off between stability and clearance.
Okay.
A CF bond is often used to block a metabolic hotspot, a position on the carbon chain where those P450 enzymes would normally perform an oxidation.
So you're protecting it.
You're protecting it, so the drug stays active longer. However, if the entire molecule becomes too stable, clearance becomes difficult, and you risk accumulation.
So chemists are trying to find the sweet spot.
They spend years fine-tuning the placement of fluorines to achieve that Goldilocks zone. Stable enough to be effective, but still having other functional groups that allow for eventual controlled excretion.
That makes so much more sense. So conversely, what if we use heavier halogens, like chlorine or bromine? What happens to stability then?
Because chlorine and bromine are larger, the bonds they form are a bit weaker. They also create a more pronounced positive charge on that adjacent carbon.
Which makes the carbon more electrophilic.
Highly electrophilic. It becomes highly attractive to electron-rich species, or nucleophiles.
Okay.
So while this is great for making new molecules in the lab, it means that in a biological system, the molecule is much more likely to react with biological nucleophiles like water or proteins, leading to instability or unintended side reactions.
So CF is for stability. CClBr is often for reactivity.
In a nutshell, yes.
Okay, here's where it gets really interesting. We move into the first set of functional groups where oxygen becomes part of the structure, but not as a carbonyl group yet. This is the simple oxygen heteroatomic section.
And oxygen, being highly electronegative, guarantees polarity.
We start with one of the most recognized groups, the alcohol, which features the hydroxyl group.
Right, the formula is simple, ROH. The naming ends in -ol, like methanol or ethanol.
The defining feature is that OH bond.
It is. Because oxygen is pulling electrons away from both the carbon and the hydrogen, the molecule is highly polarized. And that hydrogen atom is now capable of participating in hydrogen bonding.
Right, and an alcohol group can both donate and accept a hydrogen bond.
It can act as both a hydrogen bond donor, through the H, and an acceptor, through the lone pairs on the O. This is vital.
So in drug design, an alcohol group is a fantastic tool for binding tightly to biological targets because it facilitates these strong specific hydrogen bonds.
Absolutely. The hydrogen bond is one of the key non-covalent interactions that locks a drug into a receptor site. But there's a huge implication for solubility, too.
It makes things water-soluble.
Highly water-soluble or hydrophilic. The ability to form extensive hydrogen bonds with water molecules aids in distribution throughout the body's aqueous environment.
However, I remember you mentioning this group is also a metabolic weak spot, almost an Achilles heel for stability. Why is that?
The hydroxyl group is a frequent target for what we call phase 2 metabolic enzymes in the liver. The body sees the alcohol as a site that needs to be tagged for rapid excretion.
Tagged how?
The most common pathway is conjugation, often glucuronidation, where a large very water-soluble sugar molecule is enzymatically attached to that oxygen atom.
Which makes the whole thing much easier to get rid of.
Exactly. It makes the entire molecule significantly more hydrophilic, ensuring it's rapidly cleared by the kidneys.
Yeah.
So the alcohol group is a trade-off. Great for binding and solubility, but a known site of rapid metabolic inactivation.
Let's compare that to our next group, the ether.
The ether structure is fundamentally different. The oxygen atom now links two R groups, R1OR2. It's bridging two carbon chains.
It's not at the end with a hydrogen. Naming uses the oxy prefix, like methoxy group.
Right, or the foxy thing.
So if the alcohol had that metabolic weak spot, the available hydrogen, the ether seems inherently more stable because that hydrogen is gone.
That is a brilliant observation. Ethers are, in fact, significantly more stable metabolically than alcohols. While the ether oxygen still introduces polarity, and it's still a hydrogen bond acceptor, it cannot donate a hydrogen bond. There's no OH.
Which means it's resistant to those conjugation pathways that destroy alcohol.
Highly resistant.
Yeah.
Which is why ethers are used so frequently and strategically in pharmaceuticals.
When would you use one?
They're incorporated to increase stability and often to tweak the balance between water and fat solubility. You use them when you need a hydrogen bond acceptor for binding, but metabolic stability is also paramount. They are the stable, slightly less water-soluble cousins of the alcohols.
Now, for something that looks structurally simple, just a small ring, but is chemically aggressive, the epoxide.
The epoxide is fascinating. It's a cyclic three-membered ring. Carbon, and oxygen forming a tight triangle. Naming usually ends in oxide, like ethene oxide.
Why is this unique? It's the first cyclic structure we've seen with a heteroatom, and it just looks stressed.
You hit the nail on the head. That three-atom ring introduces immense ring strain. Yes. Carbon atoms prefer bond angles of about 109.5 degrees, but in that ring, they're forced into angles closer to 60 degrees. This deviation from the ideal geometry is stored energy, and it makes the ring extremely reactive and unstable.
Wait, if it's so unstable, how does it interact in the body?
Because that ring wants to break open so desperately to relieve the strain, the carbon atoms become highly electrophilic.
Electron hungry.
Desperate to react with any available nucleophile, any molecule with a pair of electrons looking for a positive charge.
Where do you find these in the body?
That's the problem. Epoxides are often formed unintentionally. They're highly reactive metabolites of less reactive compounds like alkenes or aromatic rings, and they're catalyzed by those P450 enzymes.
So an epoxide group is almost a molecular red flag that signals potential danger or even toxicity because it just reacts with anything.
Absolutely. Because they are so powerfully electrophilic, epoxides are notorious for reacting with cellular machinery they shouldn't, like DNA or proteins. This is called alkylation.
And that's bad.
It can cause DNA damage or inactivate essential enzymes. For a drug designer, an epoxide functional group is almost always avoided in the final product. Or if it's formed metabolically, the body needs specialized enzymes, like epoxide hydrolase, to rapidly detoxify it.
That was a powerful lesson. The placement of one oxygen atom OH, ROR, or in a ring can define a molecule's entire fate.
Structure, stability, and biological interaction, all perfectly aligned.
So what does this all mean? We've covered single-bonded oxygen, but the most versatile and important family in organic chemistry centers around the carbonyl group.
The C double bond O.
Right. And this is where we define 7 distinct functional groups just by changing what's attached to that carbonyl carbon.
This is truly the central hub, the workhorse of organic chemistry. The carbonyl is hyperpolar. That oxygen pulls so strongly on the electrons, making the carbon atom strongly electrophilic, electron hungry.
And the key thing to grasp is that just by varying the atom attached to that carbon, you get 7 subgroups with wildly different behaviors.
Exactly. Different stabilities, different biological roles.
Let's tackle the 2 simplest ones first. The simple carbonyls.
We begin with the aldehyde. Here, the carbonyl carbon is attached to at least one hydrogen atom, R, C double bond OH. The name ends in al, like ethanol.
Because of that exposed hydrogen, aldehydes are very easily oxidized into carboxylic acids. So are they inherently reactive?
Compared to ketones, yes, highly reactive. The presence of that small exposed hydrogen allows easy chemical access for oxidizing agents.
So what does that mean in the body?
In the context of drug metabolism and toxicology, aldehydes are often considered short-lived, transient intermediates. The body has specialized enzymes, aldehyde dehydrogenases, that are dedicated to rapidly neutralizing them.
By converting them to the much more stable carboxylic acids.
Precisely.
That rapid detoxification stands in stark contrast to the ketone, our next group. It's structurally similar but chemically robust.
Ketone features the carbonyl bridging two other carbon groups, R1, C double bond O, R2, naming ends in 1, like propanone or acetone.
Let's do that contrast again. The difference is just swapping one tiny hydrogen atom for a bulkier carbon group, but it creates two totally distinct families.
That single atom swap is everything for stability. In the ketone, the carbonyl carbon is now attached to two bulky carbon groups,
which creates steric hindrance.
Steric hindrance, yes. Physical crowding. It shields the carbonyl carbon from attack by oxidizing agents. And there's no easy hydrogen atom to remove.
So the ketone is much more stable and inert than the aldehyde. How is that useful in drug design?
It's invaluable. Ketones are much more robust. They're often incorporated into drug scaffolds to provide a strong, stable hydrogen bond acceptor site for receptor binding.
But without the high risk of rapid oxidative metabolism that aldehydes face.
Exactly. They contribute molecular rigidity and are highly stable under physiological conditions.
That clarity helps a lot. Now we move into the carboxylic acid derivatives, where the carbonyl is attached to a heteroatom like ON or a halogen, which profoundly influences its reactivity.
The parent compound here is the carboxylic acid.
This is a carbonyl attached to a hydroxyl group, an OHR, C double bond O, OH. The naming ends in oic acid, like ethanoic acid.
And the name says it all. It's an acid.
And what makes this group so critical in drug action beyond just being acidic?
The structure itself, with the CO pulling electrons in the OH bond, makes it easy for that proton to leave. It's highly polar and acidic.
And at physiological pH around 7.4?
The carboxylic acid is a strong enough acid that it's almost entirely ionized. It exists as a negatively charged carboxylate anion, RCO-.
And a negative charge is key for biological interaction, correct?
Absolutely vital. This negative charge gives you two huge advantages. First, it confers tremendous water solubility, which helps with clearance. Second, and most important for binding, this negative charge lets the drug form crucial ionic interactions, we call them salt bridges, with positively charged amino acids on the drug receptor.
And those ionic bonds are some of the strongest non-covalent interactions available.
Exactly, making the carboxylic acid a defining feature of many potent drugs, like NSAIDs.
Following the parent acid, we have the bizarrely structured acid anhydride.
The anhydrate is chemically derived by, well, removing a molecule of water from two carboxylic acids and linking them. It's two carbonyls linked by a bridging oxygen atom.
Two electrophilic carbonyl centers fighting over one bridging oxygen. That sounds like a recipe for disaster in water.
It is extremely unstable. The two adjacent carbonyls compete for the electron density of that central oxygen, making the whole structure highly vulnerable.
So it breaks down easily.
Anhydrades are highly reactive towards nucleophiles, especially water. They hydrolyze. They break down almost instantly into two separate carboxylic acid molecules.
So given that extreme reactivity, they are clearly not used in stable pharmaceuticals.
Correct. Their primary role is as high-energy synthetic reagents in a chemistry lab. They are far too reactive and short-lived to survive the human body.
Next, we have the ester, which is much more common in drugs, often as a sort of time-release mechanism.
The ester is a carbonyl attached to an alkoxy group, an OR1C, double bond, OOR2. Naming ends in oat, like ethyl ethanoate.
You mentioned that esters are a fantastic example of manipulating stability for drug function, often used in prodrug strategies. Can we go into detail on that?
This is one of the most elegant applications of functional group chemistry. Esters have this intermediate stability, more stable than the anhydride, but less stable than a ketone or an alamide.
Okay, so it's in the middle.
Right. They are stable enough to survive being swallowed and absorbed into the bloodstream. But once they're there, they are specifically targeted by enzymes called esterases, which are found in the blood, the liver, and the gut wall.
So the ester group is a built-in timer. The esterase sees it, cleaves the bond, and releases the active drug.
Precisely. The inactive ester form, the prodrug, might be used because the active carboxylic acid form is poorly absorbed or tastes terrible. The ester masks the active group, improves absorption, and then acts as a delivery vehicle that's programmed to be cleaved, releasing the drug.
It's a fundamental tactic in optimizing oral bioavailability.
It is.
Now contrast that intermediate stability with the rock-solid amide.
The amide is a carbonyl attached to a nitrogen group, usually NH2. R1, C double bond O, NH2. Naming ends in amide, like ethanamide.
And this group is foundational to biology itself.
It is the backbone of life. The amide bond is the peptide bond that links all amino acids together to form proteins.
And the reason it's so ubiquitous is its extraordinary stability.
Its stability and resistance to hydrolysis, yes, especially when you compare it to an ester or an anhydride.
Why is the amide so much more resistant to break down?
The difference is a concept called resonance.
Resonance.
The electrons within the CO and the CN bonds are shared or delocalized between the oxygen, the carbon, and the nitrogen. This sharing gives the CN bond a partial double bond character.
And that makes it stronger.
It makes it more stable and much harder for water or enzymes to attack and break.
So in drug design, if you need a structural connection that absolutely will not break down easily, you use an amide bond.
Exactly. Amides are favored when stability and resistance to metabolic enzymes are paramount. They also provide excellent hydrogen bonding capabilities, contributing strongly to receptor binding.
And finally, rounding out the carbonyl derivatives, we have the most reactive one, the acyl halide.
The acyl halide is a carbonyl attached directly to a halogen atom, X. The naming uses oil halide, like ethanoyl chloride.
And the halogen is a superb leaving group.
The superb leaving group and highly electronegative. This combination makes the carbonyl carbon ferociously electrophilic, the most electron-hungry carbon we've discussed so far.
So it reacts violently.
Instantly with water or anything with a lone pair of electrons.
So just like the anhydride, this is strictly a lab reagent, not something you'd find in a stable drug.
Correct. The acyl halide is only used to rapidly install that acyl group onto another molecule in a lab. In a drug, it would react instantly with saliva or blood, turning into the carboxylic acid and generating hydrochloric acid, which is clearly undesirable.
That summarizes the carbonyl hub perfectly. We've moved from the semi-reactive aldehyde to the inert ketone, the charged carboxylic acid, and then through that whole stability spectrum, from the unstable anhydride and acyl halide to the mid-stability ester, and finally, the rock-solid amide.
7 distinct personalities, all driven by that one simple CO core.
It's structure dictating function in its purest form.
And it shows the chemist's ability to tune stability just by changing one atom.
Moving now to the crucial element, nitrogen. It forms the basis of some of the most complex and biologically relevant structures, especially in neurotransmission. This is the nitrogen-based category.
Nitrogen adds a new dimension because it introduces basicity and often multiple potential binding sites, thanks to its ability to form 3 bonds and possess that critical lone pair of electrons.
We start with the simplest and arguably most important group in drug chemistry, the amine.
The amine is a nitrogen attached to a carbon backbone. The primary amine formula is RNH2. Naming ends in enkimine, like ethylamine. And the key feature is that lone pair of electrons on the nitrogen.
That lone pair is the source of its personality.
Exactly. That lone pair means the amine can readily accept a proton, an H+ ion, making it a Lewis base.
A proton acceptor.
Right. If the carboxylic acid was the essential acid for drug binding, the alamine is the essential base.
And just like the carboxylic acid, this basicity leads to ionization at physiological pH.
Absolutely. Because most amines in drugs have a pK high enough, they are affected by the body's pH of 7.4. The majority of them are protonated.
So they exist as positively charged ammonium ions, RNH3+.
And that positive charge is critical. It provides water solubility, and it's used to form strong ionic bonds with negatively charged sites on receptor targets. The vast majority of small molecule drugs on the market contain an alamine group specifically for this robust ionic binding.
It provides both the solubility for transport and the sticking power for efficacy. Now, moving from the single CN bond, let's look at the triple bond, the nitrile.
The nitrile is a carbon triple bonded to nitrogen. R, C, triple bond N. Naming ends in nitrile, like ethin in nitrile. This structure forces the linear geometry, like the alkyne, but the high electronegativity of the nitrogen makes the group highly polar.
How does the reactivity of this CN triple bond compare to the CC triple bond we saw in the alkyne?
It's different. While it's highly polarized, the nitrile group is surprisingly stable in biological systems. It often resists rapid enzymatic attack. It's frequently used as what we call a metabolically robust biosausteer.
Can you elaborate on that term, biosausteer?
A biosausteer is a chemical group that can substitute another group while keeping similar biological activity. So for example, a drug might need a linear polar group, but the chemist wants to avoid the basicity of an amine.
So they swap in a nitrile.
They might swap a highly reactive or easily metabolized group for a nitrile. It provides the polarity and geometry without the basicity or high reactivity. It's a trick to improve a drug's stability and metabolic profile.
That connects nicely to the enamine, which is a double bond.
The imine features a carbon double bonded to nitrogen, R1, C double bond N, R3. Naming ends in imine. Imines are structurally similar to ketones and aldehydes. You just replace the oxygen with the nitrogen.
Is the enamine bond stable or reactive in the body?
It's generally reactive in the presence of water, especially in slightly acidic environments. Imines, or Schiff bases, are frequently involved in dynamic biological processes.
But not stable drugs.
They're less common in orally administered stable drugs because the CM bond is more easily hydrolyzed back into its components than the CO bond of a ketone. They're optimized for transient, reversible biological reactions.
Next, we hit a highly unusual and often dangerous structure, the isocyanate.
This is a linear group, RNCO. Naming ends in NL isocyanate, like ethyl isocyanate.
This structure is unique because it features consecutive double bonds, N-cation. That seems like an extreme amount of tension. What does that mean for its chemical personality?
Consecutive double bonds indicate extremely high reactivity. The isocyanate group contains two strongly electrophilic centers. As a result, isocyanates are very powerful indiscriminate electrophiles. They are highly unstable.
And they react with anything.
They readily react with any nucleophile water, alcohols, or most critically, the amino groups in proteins.
So if an epoxide was a molecular red flag for toxicity, the isocyanate is even more aggressive.
Significantly more so. Isocyanates are notorious for covalently bonding to proteins and enzymes, a process called irreversible inhibition or inactivation.
Which is why you don't find them in drugs.
They're almost never found as designed functional groups in stable pharmaceutical agents because they would rapidly and irreversibly react with non-target proteins.
The final nitrogen group is the azo compound.
The azo compound features two nitrogen atoms double bonded together, linking two R groups, R1 and double bond N, R2. Naming uses the azo prefix, like azobenzene.
That NN double bond structure looks like an interesting target for metabolic activity.
It is indeed. Historically, many azo compounds were used as dyes, but in medicinal chemistry, they're recognized as groups that can be specifically cleaved metabolically via a reduction reaction.
And this cleavage breaks the molecule apart.
It does, releasing two distinct aromatic amine compounds.
Why would a chemist design a drug to break apart like that?
This mechanism is exploited in targeted drug delivery. The colon, for instance, contains a high concentration of specific bacteria that create a strongly reducing environment.
Ah, I see.
So if a drug is needed specifically in the lower GI tract, it can be synthesized with an azo linkage. The drug remains inactive until it hits the bacterial environment in the colon, the azo bond is cleaved, and the active drug is released only at the intended site.
An elegant example of using a functional group as a biological trigger.
Yes.
That brings us to our final category, which covers two critical groups that represent entirely different chemical environments: sulfur and the highly stable ring systems.
We turn now to the sulfur-based group. And since sulfur sits directly below oxygen in the periodic table, we expect some similarities to the alcohol, but also some crucial differences.
The functional group here is the thiol.
The thiol is the sulfur equivalent of an alcohol. The sulfhydryl, or SH group, formula. RSH. Naming ends in the thiol, like methanethiol.
Can you walk us through the implications of swapping the smaller oxygen for the larger sulfur atom?
When you move down the periodic table, the SH bond is longer and weaker than the OH bond. And because the bond is weaker, the proton is more easily released. This makes the thiol significantly more acidic than its alcohol counterpart.
So it's more likely to be negatively charged.
Exactly. Thiols have a lower pKa, meaning they're more likely to exist as the negatively charged thiolate anion, RS- at physiological pH.
That negative charge suggests high reactivity and a specialized biological role.
Precisely. Thiols are absolutely essential in biology. Think of the amino acid cysteine, which uses its thiol group to form structural disulfide bonds within proteins. The SH bond is also easily oxidized, making thiols powerful biological reductants or antioxidants.
What are the immediate concerns for a drug chemist when designing a molecule with a thiol group?
There are three main concerns. First, that tendency toward easy oxidation means they're often unstable. Second, Thiols frequently bind to heavy metal ions.
And third.
And third, and perhaps most immediately problematic for the patient, thiols are responsible for the incredibly potent, often foul odor of things like skunks or rotten eggs.
Right. Not ideal for a medication.
Not at all. Incorporating a thiol usually requires masking the scent or ensuring the dose is infinitesimally small.
Finally, we arrive at the stable ring structure that is foundational to vast areas of chemistry and drug design, the aromatic group.
We are looking at the arene, the classic six-membered carbon ring, usually represented by the benzene ring.
Why is this ring so structurally distinct and stable compared to a simple cyclic alkene, which would be highly reactive?
The key is the concept of aromaticity. A cyclic alkene would have localized double bonds that are highly reactive. The arene, however, has 6 pi electrons that are delocalized.
Meaning they're shared equally over all 6 carbon atoms?
They are. It's like a collective security system for the electrons.
A perfect analogy.
Because those electrons are shared, the energy of the system is lowered, which grants it tremendous stability. This stability makes the ring highly unreactive towards the kind of reactions that would break down a normal double bond.
So what does this massive stability and unique structure mean for drug discovery?
The arene group is invaluable. It provides crucial structural rigidity and, most importantly, high lipophilicity or fat solubility.
Right.
The flat, rigid nature of the ring, combined with its hydrophobicity, helps the molecule cross cell membranes and fit snugly into the nonpolar pockets of receptor sites. We estimate 70 to 80% of all small molecule drugs contain at least one aromatic ring for these purposes.
So the arene is used to ensure stability and membrane permeability. But even this stable structure has a designated metabolic vulnerability, correct?
Yes, it's stable, but not invincible. The aromatic ring is a primary target for oxidative metabolism in the liver, catalyzed by cytochrome P450 enzymes. The body's most common method for detoxifying an aromatic ring is hydroxylation.
Adding a highly polar hydroxyl or OH group to the ring,
Yes.
which transforms a lipophilic, fat-soluble ring into a hydrophilic, water-soluble ring.
Precisely. This transformation increases water solubility and facilitates the drug's eventual excretion. So the arene is stable enough to function effectively as a drug component, but it still provides a controlled, predictable metabolic point for the body to eventually clear it. It's one of the most perfectly balanced functional groups in drug design.
We have successfully broken down all 22 essential functional groups across 7 main categories.
Hydrocarbons, halogens, simple oxygen groups, the versatile carbonyls, the basic nitrogen groups, sulfur, and aromatics. The key takeaway has to be reinforcing that core objective. Functional groups are defined by their structure, the specific atomic arrangements like COOH or C triple bond N.
And understanding these patterns gives you predictive power.
Exactly. You can categorize any molecule and immediately begin to predict its behavior in a biological environment.
We saw how the CF bond ensures resistance to metabolic attack, while the CO bond in an ester is a designed weakness, just waiting for an enzyme to cleave it. This structural knowledge is the essential first step.
It's the required shortcut before you can ask how a molecule behaves.
And we also saw the crucial role of charge, the difference between a neutral ketone and a charged carboxylate anion or a protonated amine.
It isn't just a matter of composition. It's a matter of where they go and what they stick to in the body, all driven by the power of ionic attraction.
We've seen how these tiny changes, swapping an H for an OH or introducing ring strain, can create a completely new chemical entity, dictating everything from its stability in the bottle to its half-life in the bloodstream.
This raises an important question for you, the learner, to consider further. If we take all this knowledge about polarity, charge, and reactivity, what happens when you have a single molecule that contains 2 functional groups that are chemically opposed?
Like the basic positively charged amine and the acidic negatively charged carboxylic acid.
Exactly. When these 2 opposite personalities exist on the same molecular scaffold, like in an amino acid, they can interact internally. What kind of unique internal molecular interaction does this conflict create? And how does the formation of this highly charged, balanced internal structure, what's known as a zwitterion, govern the molecule's overall behavior, its solubility, and its ability to act as a building block for life?
That's the true power of knowing your functional groups.
It is.
Support us by liking and subscribing to our channel.